And thank you, Rita. And also thanks the organizing committee for inviting me. And it is my first time attending the KDD meeting. I'm probably one of very few statisticians attending the meeting. I really feel I learned a lot and from the community, and also I'm very grateful. I feel welcomed by the community. So I will share with you some of the lessons I learned and how to build an end-to-end scalable and interpretable data science ecosystem, and using statistics, AI, and domain science. So everyone knows that we face multiple challenges in the age of AI, and ChatGPT is particularly transformative and a tipping point for AI. And so many of unstructured data, as statisticians we are not familiar with, have emerged in the last couple of years, such as the text data, imaging data, and voice data. And also data become much more complex, and also the heterogeneity of the data is unparalleled. And also we learned that the traditional statistics and the machine learning method series become challenged by many of the emerging and innovative learning methods, such as the transformer, large language model, and the diffusion models. And so also, as in academia, we face additional challenge, in particular, limited computational resources. For example, like Meta has over 350,000 GPU. At Harvard, we just have a couple hundred GPU. And so this limits what academia can contribute, but also create exciting opportunities for academia as well. And in particular, we had to reposition ourselves. And this also has an impact on the fundamental research and basically done by academia and open science research, and also academia is unique in higher education mission as well. This has impact in that dimension as well. So for researchers in academia, we have to think about how we want to position ourselves, and also what we can bring to the AI table, and also how can we collaborate effectively with researchers in industry. And so to advance the innovation of AI and also advance scientific discovery. So every field, in my field, statistics, researchers ask ourselves in our field about what we can do. And so here's my thinking using statistics example. So it's really a two-way street. So first is how can we empower a particular field, our own field, in my field is statistical science, by leveraging transformative AI potential. And second, how to empower the trustworthy AI by leveraging rigorous statistical principles and also accounting for randomness and also uncertainty, and to make the AI more powerful, more trustworthy. So my talk is organized in several parts, and the first part is the takeaway is it's important for us to be open-minded and be curious, and also think out the box, and also willing to take the risk. So use the ChatGPT large language as example. So as many of you know, like ChatGPT has a huge number of parameters. For example, 4 has a trillion parameter. When the ChatGPT just came out over 1.5 years ago, my first reaction as a statistician is this overfitting? And how can this work with this huge number of parameters, much bigger than the sample size? And so AI researchers like the ChatGPT, and their mindset is very different, engineering mindset. Make it work first and try to figure out why. I think this is good lessons for us to learn. And now if one look at the deeper, it looks like there is a very interesting double descent phenomenon. So we as a statistician are familiar with the phenomenon, the phase on the left-hand side of this blue line. So we know the right curve indicates the training error. So when the number of parameters become big, one has overfitting problem, and then we have the test training data, training error goes to 0. And so for the testing error, if the number of parameters goes up, then the testing error goes down. But when it's going up more, when the number of parameters increase more, and we have an overfitting problem. And interestingly, one has this kind of transition, the phase transition phenomenon. When the number becomes super large, the model, the true model, is more likely in the model space, and then one has this double descent phenomenon. And so this is quite eye-opening. And so this tells us that it's important for us to stay out of our comfortable zone, and also it's important to learn from empirical research and then understand the theory later. And also, a lot of effort has been done in the last couple of years to try to understand the theoretical foundation of this double descent theory. And for example, this is one of the earlier papers studying the double descent theory of the linear model. And in the meantime, more effort needs to be made to understand this double descent theory of more complex model, for example, large language model. And also the other thing, and many of you know, like in many learning methods, mainly focus on prediction. Prediction definitely is important, and it's important to put at the individual level. In the meantime, we are also interested in understanding the science and also understanding the action and making the policy. And this will require one step ahead to understand how to make an inference on the population parameters. And so, for example, one question is the BRCA1 mutation, which BRCA1 mutation causes breast cancer? And also, if a woman has that one particular pathogenic genetic variant, what is lifetime risk for developing breast cancer? So if one has one particular pathogenic variant, the risk can be much higher compared to somebody without that mutation. And also, the other question is what action can be taken, and whether this is a good action or not. For particular prevention action in this phenomenon is like breast removal, and so this is mastectomy. And So this basically brings up multiple things we ask ourselves as a statistician, that we need to do something more disruptive. And also computer scientists, and they also think about something more disruptive in the computer science field as well. And first is that, and in the large language model, how can we assess uncertainty? How can we account for uncertainty? For example, if one asks a question in ChatGPT, should women with one particular harmful BRCA1 mutation have mastectomy, basically breast removal? And so if the answer said yes, but then one need to know how accurate that answer is, and then one need to provide a model-based confidence score or probability. And how can we do this? Because not every answer from ChatGPT is correct. And also, how can we use the large language model and then make inference about the parameter of interest in the population? And so this population parameter may not be a parameter of this 1 trillion parameter in the large language model. For example, suppose we use the large language model to estimate the risk of developing breast cancer after metastasis. And so when we need to know this parameter may not be in the large language model, so this is the population parameter, it's important, it's the action. And then how can we make inference on that? And so this is quite challenging right now because we don't quite understand the model. And also, we ask ourselves, and what are the data? What do we mean by data? And so traditionally, in health research, study design is critically important. Sampling is important. And then text, video, and voice data from the internet, there's no sampling frame. And how can we assess uncertainty? And then also, should the synthetic data and augmented data from the generative model used to learn the features be considered the data? And so those are quite interesting question, but a hard question. And also, what do we mean by model? Traditionally, with the statistical model, and compared to the large language model, large language model and transformer, for example, are much broader and also implicit. One cannot write it explicit model down. And also, the model parameter is mentioned of interest, and may not be of interest, but the population parameter may be of interest. They are not part of the model. And also the feature space, and so are generally not pre-specified and in those large language model and also self-learned. So to illustrate this point, if one compares the traditional machine learning with representation learning, traditional machine learning basically manually pre-specify the feature space and basically the functional space, like linear space, quadratic space, or the spline radial basis through the feature engineering manually. And for example, like random forest, and the feature will be the trees. And the kernel machines and the traditional kernel machine, like linear kernel, the feature space will be the linear space, and the Gaussian kernel, the feature space will be the radial basis. That's some kind of reproducing kernel Hilbert space. But if one think about representation learning, and the thinking is quite different. It extracts useful feature automatically from the data without pre-specifying what kind of feature space one should consider about in advance. And so, for example, if one think about deep learning, the special case of representation learning that uses a neural network with many different layers. And so that compared to traditional machine learning, one manually extract the features and through some feature engineering, but now one basically uses self-learned feature from the data. And so this also bring a question, what kind of functional space we talk about, and this will help us to understand what studies the property of the method. So if one think about formulation of representation learning, if you have the input data, suppose you have some kind of a true generative model as function of some kind of features, and so one want to estimate the data-driven feature automatically without pre-specifying the feature space, for example, some kind of reproducing kernel Hilbert space or the linear space. And so that can be used for downstream tasks. So this is quite different from the traditional statistical framework or traditional machine learning framework. And so this is takeaway number 2. And what I have learned in the last 10 years by working with large whole genome sequencing study and biobank and EHR data, it's important to be a full data scientist. What we mean by that? So that means we need to build a data science ecosystem. So from my perspective, the data science ecosystem has 3 pillars. One is the data need to be diverse and fair. And this will involve careful study design. Second, one need to develop scalable statistical machine learning and AI method, tools, and resources. And the third is to make those tools give the analysis result interpretable. And also, so in order to achieve this goal, build this type of system, data science ecosystem, and it's important to become a full data scientist and who have expertise in domain science, which include the statistics and the computer science, and also the particular domain science. For example, in my field will be the genetics and health. And also, it's important to have the communication skills, and basically one can communicate effectively with the domain scientist, and also with, for statistician, effectively communicate with the computer scientist. And so one can basically speak for the same language, and that will make the collaboration more effective. The optimal goal of doing this is for people from different fields to work together and to solve a big real-world problem to make an impact in science to advance scientific discovery. And so what I have learned by analyzing those very large data is that data fairness becomes more important than the size. And data size is right now is not the issue. So for big data, bias is much more important than the variance. So this will require increased diversity of the study sample. To illustrate, so Genome-wide association study, and so has identified tens of thousands genetic variants associated with the human diseases. And so basically one sequence a large genotype, a large number of breast cancer cases and a large number of breast cancer controls. For example, like a couple million common genetic variants across the genome, and then scan the genome and let's find out which genetic variants associated with the breast cancer. And this effort has been quite successful, but in the meantime it has an issue. 86% of the GWAS sample in the last 10 years are European. And so they're basically represented by the right part of this curve. And so you can see the samples of East Asian, African American, Hispanic much smaller. And this generally generate health disparity issues and also generate issues about risk prediction as well. To illustrate, so polygenic risk score, what is the idea of polygenic risk score? Suppose if one genotype a person, and so like several million variants of genetic variants. And then one can aggregate those genetic effects across these couple million genetic variants to estimate the risk score. So basically, use the GWAS summary statistics, and this grid creates the SNP weight. So basically like a regression coefficient beta. If a new patient comes in and use this person's genotype file and multiply by the weight, then one can calculate the risk score for that person. One can use that to do the risk stratification. And so in the field of genetics, in the last 10 to 15 years, a lot of effort has to make the summary statistic public available, for example, in the GWAS catalog or the dbGaP. So what is the issue? And there is the issue of the fairness. So because majority of the GWAS study have been done among Europeans, so if one uses the polygenic risk score weight from the European as a weight and apply to other populations, suppose that red dot is the European reference. When that applied to the East Asian, that is a blue dot, you can see that accuracy is only 50%. When one apply that weight to African American, it's only 25% accuracy. So this tells us the importance of data fairness, and also it tells us the importance of algorithm fairness. So how can one use a large sample size of European and small sample size of African American and perform transferred learning to improve the risk prediction accuracy for African American and then implement it in clinic? And so, as statistician or computer scientists, we all want to try the fancy method. For example, lots of effort has been made applying the deep learning method and to do the risk prediction to calculate polygenic risk score. However, the performance is not that great it's not that much better compared to the existing method. And so why is that? The reason is the GWAS signals, the genetic variant signal, generally are weak and also mainly linear. So if one think about the autoencoders that generally has the decoder phase that identifies the representation and the feature space, and also the decoder space, and also regenerates the data. So if you think about the simplest version of the autoencoder, like an encoder, so basically this is a feed-forward neural network. Suppose there are 3 layers, you input the genotype, and then you create the activation and using the activation function. And so this W is called the weight, B is called the bias. And then you calculate the A, and then you do that again, and as a last layer, and then basically you do this convolution. So you can see that one can estimate the W, one can estimate the B. And so, but if the F is a linear function, and so you can see there's no need to do the convolution. And so what this tells us is the following. When the effects are linear and mainly weak, and the convolution neural network or deep learning are not necessary. So what also tells us the importance of understanding the underlying science and to avoid wasting time. For example, in genetic study of common diseases, the effects are mainly weak and not much interaction. And finally, in the real world situation, biological signals for common disease are often weak. So like for images, and so for example, like we all know, like deep learning are successful in handling imaging data. The data generally signal are much stronger and the features are nonlinear. But in biological signal, most of the time they are weak. So how can we develop deep learning and also transformer method for weak signal? This is an open question. And so for our effort, we have done some work in the transfer learning to use the transfer learning tool to build the polygenic risk score, to transfer the polygenic risk score from the European population to African American. So this work was led by my former postdoc, Hao-Yu Zhang. So the method he developed called CDSLEB, and that is the dark purple curve on the right-hand side. So this provides prediction accuracy. So you can see that compared to other existing methods, and when this method, transfer learning method, applied to African American, and you can see that it's better than the existing method. So this paper was published in Nature Genetics last year. And also, translational science is critically important. In particular, how can we make polygenic risk score clinically provide clinical value of polygenic risk prediction? And so translating those kind of polygenic risk profile into the action, into the intervention, and into the primary care. And so this work was led by my student, Tony Chen, and also with the collaboration with Liu Chen at WashU. So we are currently running 2 clinical trials. And so these 2 clinical trials, one is called PRECISE, the other is called MOTIVATE. So basically, we are enrolling about 1,600 patients, lung cancer patients, and return the polygenic risk score of those lung cancer patients to the patient to see whether they are more motivated to quit smoking and also start bioco-treatment. And so in particular, these trials, and to see if one has a high polygenic risk score, and whether this will help the high lung cancer risk patient change their behavior. And so, for example, whether they will be able to quit, more likely to quit the smoking, and if they cannot quit the smoking, whether they are more willing to take the tobacco treatment. So the trial is still ongoing, and so we will have the result available in a couple years. And the takeaway number 3 is many of the health data, because confidentiality issues, individual level data are not publicly available. And then, however, summary statistics are widely available, like I mentioned, the GWAS summary statistic. So this also tells us that it's important to share the summary statistic and also really think about how we can fully leverage the summary statistic for the machine learning method without individual level data. And so to illustrate, and as I mentioned, GWAS data, the genetic data or electronic medical record data, individual level data generally not publicly available. And however, summary statistics are widely publicly available. So for example, in GWAS summary statistics, one can think about X transpose Y, the simplest one, and that's called the score statistic, or the X transpose X, we call the LD. And we all know if one knows X transpose X and X transpose Y, and one can do linear regression, one can do meta-analysis. And then the question is, can we go beyond meta-analysis? Can we go beyond linear regression without individual level data, but get a similar result to the result using the individual level data? And the answer is quite promising. So I'm going to use one example. We call the all-sum. And so this kind of penalized regression has been widely used in the prediction. And for example, using L1 penalty, we'll use the L0 penalty or L2 penalty. We call the L0 learn. And so this is a loss function using the individual level data with L0 and L2 penalty. A natural question we ask ourselves is, suppose I don't know the individual level data, Y and X, and then but I know X transpose X and X transpose Y, can we still calculate the beta hat? And so the answer is yes, we call the all-sum. So this work was led by my student, Tony Chen, appeared in PNAS 2 weeks ago. It was selected at the PNAS showcase. So in particular, doing a simple calculation, and one can show that if I have the tuning parameter, lambda 0, lambda 2, and then one can update the beta hat using the coordinate descent optimization, you can see has a iterative closed form, basically through the iterative thresholding and through iterative shrinkage. And so, but the tuning parameter, the thresholding parameter, and also shrinkage parameter, as you can see here, is the same. So in order to do this, one does not know the individual level data, one just need to know the summary statistics, X transpose X and also X transpose Y. That is great. Also, we found another thing is that traditionally to estimate the tuning parameter, we all know tuning parameter is critically important in those kind of prediction model. And then traditionally, one basically uses a grid search to find optimal tuning parameter. We found that if one uses ensemble method, one can have a better tuning parameter estimation. So suppose you have a bunch of tuning parameter lambdas, and for each tuning parameter, you got a beta hat. And so suppose you have 1,000 tuning parameter, you got 1,000 beta, so the 1,000 grid, you have 1,000 beta hat. And now the question is, how can I calculate the best y hat? One can pick to see which beta hat gives me the smallest testing error. And this is a traditional way to do the grid search. But we found that suppose I use ensemble method, I use each beta hat, calculate the predicted value. And then I calculate the kind of penalized likelihood, and then find out which combination of those predicted value give me the best beta hat. Basically, this kind of, you can see this is the weighted average of the beta hat estimated from each tuning parameter. We found this kind of ensemble method work much better. So if you look at this, you can see that basically one uses this ensemble method, instead of use the same tuning parameter, the same thresholding and the same shrinkage parameter, here use the different thresholding parameter and different threshold and different shrinkage, and to estimate the beta hat. So that's why it is better. If one compare this approach, this ensemble super learning approach, with the traditional grid search approach, one can see that basically when you use the training data, do the tuning data, and regress on each of the y hat, and then find out which best y hat, and to predict y. This basically is a grid search approach. And so basically this corresponds to one step forward selection to identify the best single y hat. So that's why you can see this method, grid search, is worse than the ensemble method to construct the prediction. So ensemble estimation is a good thing. To illustrate, we apply this to the UK biobank data. So if one uses LDL, this is a bad cholesterol as an example. So the dark blue on the right is using this awesome approach compared to the other method. You can see it has a better prediction performance. So that indicates one can use a summary statistic without individual level data to get a similar result, and also ensembling method for tuning parameter estimation provide a better prediction performance. So this is the takeaway number 4. And so to make the real world impact, what I have learned, it's important to build end-to-end data science ecosystem. And for statistician, I think we have to move one step out of our comfortable zone and try to engage in data science engineering, because the goal is to do the best science. And so basically the last mile is critically important. It's important for us to push the last mile to deliver the product. And to illustrate how to build this end-to-end data science ecosystem, so I'm going to use the whole genome sequencing study as an example. So this will involve the functional annotation of the whole genome. And so as we all know, people like to do method work, and people are less interested in doing the data work, and annotation work take a courage. But it's really important to do the scientific discovery. And so I'll explain what this annotation effort is, and then build automated analysis pipeline. So basically, if one provides me the whole genome sequencing data, one provides me electronic medical record, I can do the whole analysis without thinking about the details. And so this is completely automated. And also, we need to deploy the whole thing and in the cloud, make it scalable and cost effective. The reason is many of those large biobank data, such as UK biobank data, cannot be downloaded anymore. So it's all on the cloud. And so this is the whole genome sequencing timeline. So in the last 10 years, the sequencing cost has dropped dramatically. Several large whole genome sequencing programs have been launched in the US and also around the world. In the US, so they include the TOPMed, which is called Trans-Omics Precision Medicine Program by NHLBI. That we have sequenced about 200,000 people with many different heart, lung related diseases. The other one is called the Genome Sequencing Program, that is by the National Human Genome Research Institute. So we have sequenced about 350,000 people. And also, many biobanks have emerged. The most well-known biobank is the UK biobank, which includes GWAS data, whole genome sequencing data, epidemiological data, and electronic medical record, and which has about 500,000 people. And the UK biobank has made the data public available to the community. Everybody can apply for access. And but I mentioned the data cannot be downloaded anymore. It is only on the cloud. And the other one called the Million Veteran Program has enrolled 1 million people, about 100,000 people. And so the GWAS data has been done and for 1 million people, but also whole genome sequencing data are being generated. The other one is called ALPHAS, and so the plan is to enroll 1 million people. Right now, probably has enrolled about 800,000 people. So ALPHAS is in the US. It used to be called the Precision Medicine Program. So this where this term comes from, and announced by Obama administration. And then later on, and so this the name is changed to ALPHAS. The idea is to increase diversity of the sample. And so why biobank? So biobank, a major benefit of a biobank is a one stone hit multiple birds. So traditional, when one does a genetic study, one focus on particular disease. For example, breast cancer, sequence like 10,000 breast cancer cases, 10,000 controls. So spend so much time doing the genotyping, but only analyze one phenotype. But using the biobank, one can look at thousands of phenotypes and from the electronic medical record. And so therefore, it's much more effective. And so in terms of the cost. And so the UK biobank and the whole genome sequencing data are currently available in the UK biobank platform, cloud platform called the RAP, and also the cost largely covered by several biopharm companies. The reason why the biopharm company are interested in doing this is that for clinical trials, and from phase 1 to phase 4, and the success rate is about 10%. So you can see why the drug is expensive, because clinical trials generally quite expensive and only 10% success rate. However, if a drug target has the genetic basis, then turns out the success rate is higher. So that's why biopharm company is interested and in investing in the sequencing effort in those kind of large biobank. And also in the last couple of years, there are dramatic efforts to increase the data fairness and in the large biobank and whole genome sequencing study to make them more diverse. And so like you can, so for example, if you look at TOPMed and the Genome Sequencing Program and all of us, they are all multi-ethnic, have a good representation from European, African, and AMR and Asian. So if one think about a lot of those kind of online data, and it's difficult to increase this kind of data diversity without designing a study. And so therefore, kind of study design is critically important and to increase the data diversity. And so the data we deal with are humongous. And so for example, for the TOPMed data sequence about 200,000 people, before the QC, they are about 1 billion variants. After the QC, they are about 845 million variants. And many of those variants are rare, and so that's why humans are different from each other. For example, 45% of the variants are singleton. That means only one person has it. And so 16% doubleton. That means only 2 people have it among the 200,000 people. And so what is the goal? We are not interested in this situation, trying to see whether the whole genome is associated with breast cancer or cardiovascular disease. What we want to do is, suppose we want to scan the genome, identify the genetic variant set associated with the disease, and then those can be identify the potential drug target, and also then this can help develop the precision intervention, precision medicine strategy. For example, if one has a particular mutation, and then immunotherapy or the targeted therapy will be effective. So therefore, it's really important to understand the science. And so the inference is equally important as the prediction. And in order to analyze the whole genome sequencing data and to identify which genetic variants associated with the human diseases, identify potential drug target, an important step is to do the genome annotation. Functional annotation basically try to understand the function of every particular variant across the genome. Each person has about 3 billion positions across the genome. At each position, there's one reference allele, 3 alternate allele. So totally, there are 9 billion genetic variants across the genome. So we want to understand every position functional information. And this is also called the site information that can be used for the whole genome sequencing analysis and also to particularly identify potentially position or potential variants, and biological experiment can be done to understand the biological function through the experiment. So to do this, and through the whole genome sequencing program, and we developed the FAVOR, this is a functional annotation database of 9 billion variants, by integrating functional data from many, many different sources, from ENCODE, roadmap, and so on. And so this includes allele frequencies, conservation, protein functions, epigenetic function, and so on. So you probably heard about alpha missense, and so that by the DeepMind group. And so we have, that basically predicts the functional information of a missense variant. So we have incorporated those type of prediction and in those large databases as well, and make that public available. And also, many researchers are not specialists, and so they may not be computational biologists, and so to help improve the user experience, and we also developed with FAVOR GPT, and so basically use the ChatGPT API to develop a FAVOR API, basically use this interface to query the backend FAVOR annotation database that create a convenient large language model interface of FAVOR. So this work was led by Tom Lee, who is a genius high school student, and so he's a high school senior. Sometimes it's really wonderful working with some smart high school student, and also my computational biologist, and also my software engineer. And to illustrate, so for example, like if one asks, just like what you do in ChatGPT, you ask, provide the functional information of this particular virus, and then that will use this RAGs and use the FAVOR backend database to provide a summary of this functional information and in English, and make it easier for people to understand. And also can do all kinds of calculation, use this large database. For example, if one asks how many pathogenic variants EBRC1, then it will provide totally 92 using the ClinVar and the database. So you can see that one can do all kinds of calculation and then use this high quality, specialized database and integrate with this kind of large language model, this large database. And then the query information will be much better compared to use a large language model. And also, in order to do the analysis, it's important to see how we can work with the cloud, and then build this data science ecosystem, use the cloud-based data sharing and computing. The traditional way with the data sharing, basically data copy, for example, for UK biobank data, one can download the data in the past, and two individual clusters, for example, I can download one to Harvard cluster, other can download to your own cluster. So the data sharing basically the same as the data copy. There's no sharing of the infrastructure, and basically bring the data to the researcher. And so then, but now basically move to bring the researcher to the shared data. And so this basically, all the data in the cloud. And so the different cloud system people are familiar with, like Google Cloud, Amazon Cloud, one need to work with in those kind of data ecosystem and work with the workspace. So NIH has made a lot of effort establishing the data science ecosystem. For example, Anvil, which hosts the Genome Sequencing Program data and also AlphaS data are in Anvil. The data cannot be downloaded in the cloud. And also Biodata Catalyst, this is by NHLBI, hosts the TOPMed data. And UK Biobank data cannot be downloaded. It's in the cloud, in the wrap. So cloud sounds wonderful, but it is expensive. It can easily break the bank for research in academia. So therefore, the cost is the important factor for us when we consider our analytical tool. And so for so for example, for UK Biobank, about 200,000 whole genome, if one uses a VCF file, and so you can see they totally have about 60,000 files, and the storage is about a couple hundred terabytes. That's quite expensive. Each terabyte, $300 a year, and it's hard for the academia to afford. So first thing we need to do is to develop an efficient data storage, and so we developed this called annotated GDS format. So GDS file has been used for gaming purposes. So we added the functional annotation to the genotype file. So annotation and the genotype file is in one file. This makes it much easier for data analysis, downstream data analysis. And also the storage, you can see that 1,000 times smaller. It's only costs peanuts. So this is really much better. And also, as I mentioned, we want to make the whole thing automated. And so if researchers provide the whole genome sequencing data from, for example, from UK Biobank, and the phenotype data from the electronic medical record, and then the whole system, and take care of the whole analysis. So we have this backend functional annotation database, Favor, that is, we made that available in the cloud system, ecosystem, like Anvil, Biodata Catalyst, and also the RAP, UKB RAP. And one can first run the QC, and to QC the genotype file, and then we have this Favor annotator, which is a script which can be used to annotate any whole genome sequencing study. For example, UK Biobank data or TOPMed data and GSP data that create functionally annotated whole genome sequencing data that can be ready for analysis. And by integrating the annotated whole genome sequencing data with the phenotype EHR data, one can perform the whole genome sequencing analysis using the STAR pipeline. And so you can see this whole thing is automated. And so how can we make the analysis interpretable? So that also means that it's important to develop a scalable and interpretable statistical and machine learning method by incorporating domain science knowledge and in the method. To illustrate that, I'm going to illustrate how to perform whole genome sequencing real-world association analysis by incorporating functional annotation. So we developed the STAR, and so this work was led by my former students, and 2 papers with Nature Genetics and 1 paper in Nature Method. So for quantitative scientists, it's important for us not only publish in computational or statistical journal, it's important to publish in Nature Science and Cell as well. And so they do publish method paper, and so to increase the impact of the work. And but the writing style is very different. So the main idea is to empower the real-world association test by dynamically upweighting the variance with the functional information, with the multidimensional function. And so the basic idea is if one has a genetic variance and knows the genetic function and also the phenotype is like, for example, like a LDL or cardiovascular disease. So one first want to incorporate the functional information in the analysis to empower discovery. And second, also it's important to understand the mechanism. So for example, whether this particular genetic variance and regulate a particular gene and then affect the disease, for example, cardiovascular disease. And this kind of understanding of the biology is critically important. That can help the action. In particular, that can help identify the drug target. So you can see this is not quite just a prediction problem. And so the inference is equally important as a prediction. And also this can help with developing prevention strategy, developing intervention strategy, and develop a precision intervention strategy. So for example, a treatment strategy as well. So therefore, for today, I'm going to just focus on the how can we incorporate functional information in discovery and in the analysis. And so definitely causal inference is critically important. For example, causal mediation analysis to help identify the causal pathway. So to illustrate, first let me introduce the genome. So each genome has 23 chromosomes. Each chromosome has a gene and an intergenic region. And for each gene, there are multiple parts, like exon, intron, exon called the coding regions, and also the promoter, and also the enhancer. And also, what we mean by the genotype, basically across the genome, they are just letters, ACDG letters. And so the genotype generally are AA, AB, or BB, one from mom, one from a dad, and the B is called the man allele, and code generally as 0, 1, 2, the number of man allele. So basically you have a lot of numbers, 0, 1, 2 number across the genome. That's basically the genotype. And now what we want to do is we want to scan the genome and to see which genetic variants associated with human diseases, for example, LDL, and this bad cholesterol or cardiovascular disease. And so in the GWAS study, generally because the variants are common, one analyzes one SNP at a time. This doesn't work for whole genome sequencing study. And whole genome GWAS study only occupy 1% of the genome. But whole genome sequencing study basically sequence every single base pair across the genome, and so basically the whole genetics code of each person. So then we need to perform the SNP set analysis, but then how to create the SNP set. And then this will need the biology information. And for example, if you think about ApoE, and suppose there are 100 variants in ApoE, we want to see whether the ApoE variants enhancer are associated with LDL, this is bad cholesterol. Suppose you have like 100 beta to do this kind of a regression, and suppose only 4 variants or 4 beta not equal to 0. In order to see whether the ApoE enhancer associated with LDL, ideally you want to do 4-degree freedom tests instead of 100-degree freedom tests. In reality, we don't know there are 4. We don't know where they are. So this strategy does not work. But I also don't want to do 100-degree freedom tests and for this beta equal to 0. So what can we do? So it's basically we need to aggregate individual variance association tests in a SNP set to improve the power. And to do this, as I mentioned, that first we need to create the mask. And so this is where the biology needs to come in to understand the biology. So we group the variants for each gene. For ApoE, group them into loss functional variants, missense variants, synonymous variants. And so for example, alpha missense, it predicts the functionality of missense variants across the genome. And also for the non-coding variants, group them into promoter and the enhancers. And also one can do the sliding window approach to cover the whole genome as well. A challenge to analyze the non-coding regions, and basically when we talk about functionality, they are generally tissue and cell specific, so that also asks for more data integration, and by integrating the single cell data and also the CRISPR gene editing data. So you can see when one does the integration, one needs to have a scientific problem in mind to do the integration to help understanding that scientific question. And so how to do this then? So about 15 years ago, and one developed the burden method. So for example, if you have the association and basically the X transpose Y, that's called the score statistic. For ApoE, there are 100 variants, one score statistic, one aggregate them. So you can basically use a linear aggregation, and this is called the burden test. And so this will work out if the effect beta are all in the same direction. And, but if the beta is like, for example, loss functional variance, this will work out well. But for the promoter and enhancer, the effects are more in different direction. So this approach will not work well. So about 15 years ago, we developed this called the SCAT method, and based on kernel machine. And so this method turns out the linear kernel work quite well. And so when one uses a linear kernel, it become very simple formula, and basically it's a quadratic test statistic, aggregate individual quadratic test statistics. And so you can see if the effects are in different direction, beta positive and negative, and the signal won't cancel out. So this is particularly useful for studying the enhancer and the promoter. And but then what should be the W? W should be the weight. Ideally, if I know there are 4 beta not equal to 0, I put weight 1 for those 4 beta, put the 0 for all the others. But in reality, we don't know there are 4 beta equal to 0. So what can we do? That is where the functional information comes in. We use the functional information as a weight. If the variance is more biologically functional, I give it more weight. But then there are many, many different annotations, as I mentioned. There are tons of tons of functional annotation. Which weight should I use? I can use one weight, for example, conservation as a weight to get a p-value. I can use a protein function as a weight to get a scattered p-value, or I can use the epigenetics function as a weight to get a scattered p-value. If I got different p-value, I see which p-value is the smallest, I report that one. That is cheating. That will increase the type error rate. So how can we account for the fact you have multiple look at the data? You don't know which weight is the best. And so my former postdoc, Yao Wu Liu, developed this ACAD method. It's a wonderful method to combine correlated p-value. This can be useful in many different contexts in your own field. And when you don't know which test to use, you try different tests and then combine them using the ACAD. And so basically, it's very simple to calculate. You have the p-value, you do the time transformation, that become a Cauchy distribution. And you sum them over, and still become a Cauchy in the tail. And no matter what correlation you have, because you have a different test, all the tests are correlated because they apply to the same data. Doesn't matter. And the tail still follow Cauchy. So this is really nice. So what this means that this can be used to combine the p-value of a different test using different annotation weight. So this is really wonderful. And so just to give you an example, so for example, you can use the manual frequency as a weight for a variance which is more rare, give it more weight. And or you can use ACAD or the epigenetic score or conservation score as a weight. For each weight, and you can calculate, for example, the scattered p-value, and you can then you can have many different scattered p-value with different annotation weight. And then you do a Cauchy transformation that become a Cauchy distribution. You combine it. This is called the STAR method. The computation super quick, take less than 1 second. Scalability is critically important to us. And so this also great. So this compared to the minimum p-value test. For the minimum p-value test, that is a test statistic, not a p-value. But in order to figure out that p-value, one need to know the correlation. That's definitely difficult to use. But the performance ACAD is similar to the p-value, minimum p-value test statistic, but automatically account for the multiple look at the data. So that is great. And so now we have this pipeline. We input the data from the genotype data and phenotype data. We create, then we use the favor annotator to annotate the genome. And after we annotate the genome, create an annotated whole genome sequencing data of the UK biobank. For example, we define the mask for each ApoE, for example, gene, have loss functional variance mask, promoter mask, and so on. Then we use a different weight and to calculate the scattered p-value and the burden p-value, and then use ACAD to combine again. By the end, we have the association of the each mask of the gene, ApoE, and the LDL. So you can see the whole thing is automated. And so the results are quite convincing. So for example, when we analyzed the TOPMed 70,000 lipid samples, and so multiple masks showed up, they all make sense. For example, loss functional variance of the PCSK9, there's already a drug associated with the PCSK9 approved by FDA, and NPC1L1 already a drug approved by FDA target towards this gene, and then they are found to be associated with LDL, and also multiple promoters and enhancer like LDLR and PCSK9 also associated with LDL. So the results are quite encouraging, and so that is great. So this also allows we identify a few novel loci as well. So this paper was published in Nature Communication last year. So as I mentioned, many of the large biobank data, they are not available for download anymore. So they are all on the cloud. So therefore, the scalability is critically important to us. For UK Biobank, for example, it's the 200,000 whole genome, and use a favor annotator in the RAP cloud platform, costs only $25, and fit the null model, 25 cents, to scan the whole genome, and also $25. So, but when one do the annotation, is one-time effort, and then you can apply that to many different EHR phenotype. So therefore, analyzing the UK biobank data, you can see very affordable. So this is very important to us to make it affordable to the community, So to summarize this part, and we provide Favor and Favor GPT that provide a comprehensive whole genome variance functional annotation database. It's a lot of effort, but really worth it. And so that for all 3 billion positions, 9 billion variants, and also we provide this Favor annotator and can functionally annotate any whole genome sequencing study like UK Biobank, ALPHAS, and TOPMed, and also by dynamically incorporate the functional information annotation in the analysis method, and we develop this star that can empower the real variance association test. So this is the last part and about ensembling. So I showed earlier, like when we do the prediction, ensemble estimation with super learner is a good thing. And also ensemble method is a good thing and for both prediction and the inference. And so the goal here, I'm going to introduce how can we move from ensemble prediction to ensemble inference. And so this is the motivation of this work. It comes from the whole genome sequencing study. So the scat and burden method developed about 10 or 20 years ago. And so we asked ourselves, are these the best method? They are very, very popular nowadays, and so being widely used in whole genome sequencing study. But we challenge ourselves, and is the scat method the best method for analyzing the non-coding region? The answer is no. And so, for example, we asked ourselves, is the burden method the best test if the beta are in the same direction? The answer is no, because for the burden method, and assume all the beta to be the same, but in reality, among those 100 betas, and for example, in the loss functional variance of ApoE, even the betas are all positive, but they may not be the same. And so then we ask ourselves, can we develop a powerful test, assuming the effects are all in the same direction, but the value may not be the same? So if one performs this type of test, you can see this is difficult to do because alternative is constrained. And also the dimension is not small, can be 100 or 1,000. So let's see how can we do this. And so let's first review ensemble prediction. A simple example of ensemble prediction is random forest. So you can see for random forest, so basically one does is you have the data, you generate 1,000 bootstrap samples. And then for each bootstrap sample, you create a tree. And so most of the time, when you create a tree, and you don't want to use all 1,100 variables to create the tree, you use 10 variables, take square root. Why you want to do that? The reason is, if you don't do that, all the tree will be correlated. When you sum them over, the variance will be too big. The prediction has a bigger variance, the performance will not be good. But when you do these random samples of 10 variables at a time, then the tree becomes more independent. Then the prediction variance will be smaller, and so that will be better. And so after you generate 1,000 trees, and then you average them, this become random forest. This is just a very simple example of ensemble prediction. So is this better than bagging? Yes. Bagging basically use all 100 variables and to build a tree, and all those trees will be correlated. The variance is too big. And so now the natural question for us is, given the success of ensemble prediction, and how can we do ensemble inference in particular, and how can we do ensemble method for testing? So let's review the random forest ensemble learning. So here, suppose we have a base learner, and so this kind of concept applies to other learner as well. So you have a bunch of base learner, and so in the random forest, the base learner basically will be the tree. You do a bootstrap sample and generate lots of random trees. And so each tree is a weak learner. And you use each tree to do a prediction model. But when you assemble them, average them, and this become a stronger learner, this become the random forest. So that is great. The question is, how can we construct ensemble testing? So basically, you can see what we need to do is basically create a bunch of base tests. And for each base test, we calculate the p-value, and then aggregate the p-value. Then the question is, how can we create a base test? So what we are going to do is we are going to introduce the random weight to do the random projection. and don't create a base test. Many of those random weight will be wrong. And so therefore, the p-value, many of those p-value are likely to be large because those random weight will be wrong. So if we apply the same average idea, will not work. That's basically you average them, and then that will, many of those wrong weight will dominant. But as long as there are few random weight close to the true signal direction, and I want to use those weight. So that's why when we ensemble those correlated p-values, and we are going to use the ACAT method instead of averaging, because ACAT method performs similarly to the minimum p-value test and try to identify that weight closer to the true signal direction. This basically becomes ensemble test. And so to illustrate, so for example, the Oracle test, suppose I know the true signal direction, the true beta. I project the score statistics and to the true signal direction. And so this is optimal one. That is the right line here. Suppose all the beta are positive in the first quadrant. If we use a burden test, that is 45-degree line because assume all the betas the same. That's not good. And so now what is the ensemble testing? Ensemble testing, basically, I generate a lot of random directions, those green lines, and in the first quadrant, lots of them, do 1,000 times. Many of those green lines will be the wrong direction. As long as I have 1 or 2 green lines and in the correct direction, close to the true signal direction, I'm good. Then basically ACAT method will pick up that direction and calculate the p-value. So this is quite powerful. And so therefore you can see ensemble method is really, really convenient and is quite robust. So basically, you can see you generate lots of random directions. In the first quadrant, you project your score statistics X transpose Y and to that random direction, and many of those directions will be wrong. It's okay. How can we generate the random direction in the first quadrant? Simple. You generate the Z from normal 0, 1, you standardize it, it becomes a polar system, and then you do that 1,000 times. And then after you do that, you generate 1,000 p-value. Many of those p-values are wrong, are big p-value because the direction are wrong, and also all those p-values are correlated because they are applied to the same data. Okay, no problem. And because when I use the ACAT method to combine, basically ACAT method is very similar to the minimum p-value method, identify that direction close to the true signal direction. And so this will be quite powerful. And if you have a functional information, and then instead of generating the Z from normal 0, 1, you generate it from normal 0, A, that is functional information. So that's how you incorporate annotation and in the test as well. How does this work? And that worked quite well. So if we apply this to the Eric whole genome sequencing study, and so it's identifies the genetic regions associated with the lipoprotein, for example. So you can see if I use the burden method, if I use ensemble burden, the number of discovery increased by almost 10 times. If we use the SCAT method, you can see comparing the SCAT versus ensemble method, it increased 50% time. So this is quite ensemble ideas, quite useful for not only the learning, but also for inference, for testing and estimation as well. And so basically, to summarize this part, ensemble test is a flexible and a robust way and to boost the power of the weak base test. And so in ensemble test, it introduced the random weight in the base test and also ensemble them and using the ACAT. To summarize, and so it's important for us and to be open-minded and willing to take the risk and when embracing AI in our own field. And also, it's important to improve the data and algorithm fairness. And not everything can be done by AI. And so the humans are important in the study design. And for example, improving the data fairness and algorithm fairness. And also, it's important to be a full data scientist and knows computer science, AI, and statistics, and also domain science, and then can be effectively communicate and in the different community. And also, it's important to build a scalable and end-to-end data science ecosystem, and using statistics, machine learning, AI, and domain science, and deploy it in the cloud. And also, it's important to increase to integrate the domain science in the method development that will help make the method more powerful and also to better for scientific discovery. So this is the last remark. So it's important for all of us to really work together. And so as a computer scientists, statisticians, and domain scientists, and academic researchers, and also industry researchers, and also moving forward together. Thank you.